different ages, and you can see how their temporal characteristics changed with age. For example, latencies and delay between positive and negative components, but their spatial characteristics were not dependent on age. And here you can see the distribution of locations of negative peaks relatively to the positive peak, this orange dot. So they are located caudalaterally. Okay. And also we checked if these responses are indeed of the auditory nature. To do so, we compared them with responses to electrical stimulation of subcortical structures. For example, stimulation of the cochlea. So you can see here that the pattern of activation is quite similar, and the same holds for the stimulation of the inferior colliculus, which is auditory midbrain. So responses are approximately at the same place, and also after the onset of hearing, we were able to obtain responses in the similar location, responses to the air-conducted sound. And all these results confirm the auditory nature of these responses, and also they were eliminated by the cochlear ablation, bilateral ablation. So these cortical responses have indeed cochlear origin. So finally, we compared different developmental events, and we provide here the timeline of auditory system development, combining previously known results from morphological studies and from studies, for example, spontaneous activity with our data. And we can divide 3 periods of development. The initial period of absolute deafness, then the latent hearing, when there are no responses to normal environmental sounds, but it is still possible to evoke auditory responses in the cortex using one conducted sound, for example. And after that, at the end of the second postnatal week, we see the onset of normal hearing, as we usually think about it. And another interesting thing was that this developmental timeline is quite similar to the development of the visual system. In the visual system, first responses can be also obtained from the 8th postnatal day, and there is a significant change at around the end of the second postnatal week between the activity profiles. So overall, this system demonstrates striking similarities in their development. So to summarize, we found that one conducted sound evokes thalamus-mediated cortical responses from P8. We found that a positive component represents thalamic input and negative component intracortical propagation. And we confirmed the auditory nature of these responses and found the striking similarities between auditory and visual system development. So Thank you for your attention. And also, I'd like to thank my colleagues.
Thank you, Roman. Thank you so much.
Thank you. Yeah. If there are any questions, I'm ready to.
Yes, if you have a question, post on question, I can read the question. Thank you so much for interesting study on the hearing. How big is this bump that you're showing on the slides compared to the size of the single neuron in the auditory cortex?
So it is mesoscale imaging, so we cover quite large area. For example, here you can see the scale bar is about 400 microns. So the overall area is several millimeters.
Oh, so it's quite big. It's like,
yeah. So we can see here the entire auditory cortex and some adjustment regions as well.
Right. Since in visual system, like for example, if you see the Starbucks neurons, like they develop, the size dramatically change over the P0 to P10, P11, and I believe it's supposed to be the same in auditory.
Yeah, so probably on different scale, we will see different pattern.
Awesome. Thank you a lot, Roman.
Thank you.
That was a great talk. I want to introduce the next speaker, and our second speaker is Silvana Vacha from New York University. Silvana did her graduate work with Dr. Laurent Vannes at Collège de France in Paris, working on spark timing dependency plasticity. And in 2017, she joined the laboratory of Dr. Robert Fromke at NYU and focusing on the synaptic mechanism and neural circuit important for material behavior in mice. Her research is supported by the Leon Levy Foundation and NIRSAD, Young Investigator Awards. I think that all people know about the caregiving of child and the mother, but when I read the abstract, that was fascinating to know that they, based on her work, she's digging into more connection between them when they are pups, and she's looking at the auditory pathway that way, and how does it change the whole developments. I'm very much looking forward to our talk, and Sylvia, this is your stage, and you can share a screen with us.
Thank you, Robert. I want to enable my video, but it's disabled by the host. So I'm wondering if it's maybe some sort of setting that you have.
Sure, sorry, that is probably something that I can fix it here. I will try to fix it. You can go ahead, Silvina. I will tell you when I fix it. Thank you.
Okay. Yeah. Thank you. Thank you for everyone for coming. Thanks for moderating the session. So now we're going to talk a little bit about how infants use vocalizations to attract caregivers. So we know that maternal behavior is critical for offspring survival. And one of the most well-studied forms of maternal care is the nursing itself. So it's well known that nursing relies on the signaling of the neurohormone oxytocin. So oxytocin is synthesized and released from the maternal hypothalamus during suckling, then it gets into the blood to facilitate the milk letdown. And that happens in response to tactile stimuli from the nursing infant that get relayed through spinal cord and brainstem circuits, then to eventually reach the hypothalamus. However, very curiously, milk letdown can happen in the absence of a nursing infant. And that actually happens in humans. So human mothers report that they're able to experience spontaneous milk letdown only by hearing the sound of a baby cry. And that can be their own or others' infant. That tells us that auditory cues, and specifically vocalizations from infants, are extremely powerful stimulus in activating circuits that are classically involved in nursing. So there is some sort of a crosstalk between hormonal relays and auditory stimuli. So, however, how exactly auditory information can reach the hypothalamus and then activate hypothalamic neurons, can vocalizations induce oxytocin release, is something that we're going to look at for the rest of the talk. In our lab, we work in mice, and mice are very handy because they exhibit this very stereotypic, well-known pattern of behavior known as pup retrieval behavior. Briefly, what happens is that when a mouse pup is isolated from the nest, it will cry, it will emit these ultrasound vocalizations known as pup distress calls. You can see a spectrogram of one such call here. They will track the mom and, oh great, I have video, and she will approach the pup, pick it, and bring it back to the nest to safety. So we started by conducting cell-attached and whole cell recordings from oxytocin neurons in maternal mice. So here, DAM means actually a maternal mice. I'm going to refer to it for the rest of the talk as DAM. We're recording oxytocin neurons from the PVN. PVN is the paraventricular nucleus of the hypothalamus, and our animals express channel rhodopsin under the oxytocin promoter. So by having an optical fiber in the patch pipette connected to a blue laser, we can identify oxytocin cells, because when we shine brief pulses of blue light, they will exhibit these very reliable spiking responses. Whereas the rest of the hypothalamic cells, they'll basically remain silent. So what we do is that we have a library of pre-recorded pup distress calls from isolated mouse pups. Then we loop them in a sort of a podcast, and we can play them via an ultrasound speaker to the dams while we record from their PVN. And here is what we found. I'm showing you here a cell-attached recording from one identified oxytocin neuron. As you can see, those neurons fire at very low frequencies. So this is just a spontaneous firing rate. What we do is that we play a sequence of pup calls. In this case, these are 15 calls, and then we record a second baseline. And what we found, very strikingly, is that oxytocin neurons increased very dramatically their firing rate following the playback of the calls. And you can see in this example, particularly, we have an increase of about 4 to 5-fold. And even more strikingly, we did not find that effect in other hypothalamic cells. So this is a non-identified oxytocin neuron. And as you can see, even after we play the pup calls, the firing rate doesn't change. And here are the histograms of these two cells. What we see at the population level is very similar. Oxytocin neurons, their firing rate peaks at different times following the calls. But we did not observe that in other cells. And that tells us that there is some sort of cell type specificity of auditory information being processed in the hypothalamus. So then we decided to look at what auditory areas project to oxytocin neurons. We started by injecting cell type specific rabies in oxycream mice. So these rabies will jump one synapse backward and will stain all areas actually projecting monosynaptically to oxytocin neurons. So we're specifically interested in auditory areas. And to our surprise, we did not find any direct projections from classical auditory relays, such as inferior colliculus, auditory cortex, or the medial geniculate. But we found extremely dense projections from the posterior intralaminar thalamus, or for the rest of the talk, I'm just going to call it the PIL. So PIL is a part of the non-lemniscal auditory pathway. we wanted to actually look more into depth about how this circuit is constructed. So we performed a series of optogenetic experiments in slices. So basically what we're doing here at each step is to inject channel rhodopsin in an area of interest, then slice acute brain slices of a target area and check optically evoked responses. So we found out that inferior colliculus project very reliably to the PIL, but we found less dense projections from the auditory cortex. So these are corticofugal projections. And most importantly, the PIL projects very reliably to oxytocin neurons, meaning that the thalamus to hypothalamus pathway is not only anatomically present, but is also functional. So the PIL is able to activate oxytocin cells. However, the biggest question here is how this very delayed persistent firing in oxytocin cells arises. So these responses do not seem to be classical auditory responses. They're not locked with stimulus presentation. They're too slow to be due to synaptic integration. So we hypothesized that there should be some sort of rapidly induced long-term plasticity happening during the pop call playback that will increase the output of oxytocin neurons. And indeed, we found that if we optogenetically stimulate thalamus in acute brain slices such that we mimic thalamic activation that occurs in vivo during pop calls, just optogenetically in slices, we saw that synaptic excitation onto oxytocin neurons stays the same, but inhibition drastically decreases. And that leads to decreased inhibitory to excitatory ratio, which in our case lasts to up to 20 minutes. And we hypothesized that this disinhibition of oxytocin neurons triggered by thalamic firing during the pop calls can actually induce this increase in firing. Then we perform a sequence of other experiments, and we show that this disinhibition relies on postsynaptic internalization of GABA receptors, which is mediated by postsynaptic NMDA receptors, and is dynamo independent. So now we know that NMDAs require not only presynaptic glutamate, but also postsynaptic depolarization. So right now we're conducting some wholesale recordings of oxytocin cells, looking at their depolarization, which occurs during pop calls, suggesting that they can actually be activated in vivo. And this is something that we do not see in other cells. Very briefly here, we found that if we inhibit the thalamus to hypothalamus connection with inhibitory dreads, moms will retrieve crying pups at the same rate. They'll pick up the same amount of pups, but way slower. So they actually wander around and seem to not be very motivated. Talking about motivation, we wanted to see if pop calls can induce oxytocin release in motivational areas. So we checked in the VTA. We used this newly developed genetically encoded oxytocin sensor, which is basically a mix of 3 viruses and an optical fiber implanted in the VTA. So the sensor gets activated by blue light. So if we present pop calls paired up with blue light, we can see in red all cells in VTA expressing the sensor, and in green all cells that actually have detected oxytocin during the calls. And this is our working model for the moment. I show you that indeed hypothalamus can receive auditory information, and that might be important for speeding up maternal behavior and triggering oxytocin release in downstream areas. And by that, I will just acknowledge past, current members of the lab. I certainly don't list everyone, and a lot of people from NYU and outside. And I'm very happy to take questions. Let me stop sharing.
Thank you a lot, Silvana. That was a great talk. Nicola asked a question that did you have any control sounds like a scrambled pops calls or similar?
Yeah, I didn't try scrambled pop calls. I tried pure tones actually. So I tried, single pure tones just to see if there's some sort of preferred tone in oxytocin cells, as in auditory cortex, there's none. I also tried a sequence of pure tones, just looking if the baseline firing will increase, and actually I don't find that. And that can be due at different things. So in thalamus, you have cells that respond to tones, but not to calls, and vice versa. Do they project to different targets? That can be one thing, inherited upstream. Or maybe the pure tones are not able to activate thalamic fibers in a way that, it will trigger NMDA activation.
Yes, thank you. If there's a question, you can ask in Q&A or the chat box. Okay, otherwise, I will start off by the last speaker. Great. So for a neuroscientist, songbird may not be just beautiful. We are mysterious. It's like a mysterious signal generated by brain. If your orchestra is built of many instruments, in songbirds, the analogy might be singing together or duets. Today, Santiago is talking about the relevant topic about synchroning cortical neurons. Santiago is a postdoc fellow from Institute de Física, Buenos Aires in Argentina, and physics department at the University of Buenos Aires. And his title is Rhythmic Synchronous Features in Cortical Neurons of Songbirds. Thank you, Santiago, for joining us. And I really apologize for the video. The video is working right now. You can turn on your video. Okay. And you can share your screen and start your talk. Thank you.
Thank you. So, hello, everyone. I am Santiago, currently a postdoc at Dynamical Systems Lab in the Physics Department of the University of Buenos Aires. I wanted to thank the organizers for the opportunity to give this talk, in which I will address rhythmic responses in cortical neurons of songbirds. So, as you may know, songbirds are approximately 30% of known bird species, and they are a well-established animal model to study the learning, production, and neural coding of a complex vocal behavior. Here are two examples of songbirds that we study at the lab, zebra finches and canaries, which will be the main focus of today's talk. For that reason, let's take a look at the typical canary song. Here on top, we have a song spectrogram, which is a representation of the temporal evolution of the spectral content of the vocalizations. The y-axis is the frequency, the x-axis is time, and the intensity of each frequency is represented as a grayscale. Canary song is characterized by a sequence of song phrases, which are the rhythmic repetition of certain song syllables. From earsac pressure recordings, which I'll show here at the bottom, during singing, we can see that different syllable types correspond to different pressure gestures, which allow us to classify canary song repertoire into these 4 syllable types that I show you here, which we call P0, P1, P2, and pulsatiles. Moreover, these gestures correlate with the syllabic repetition rate, such that from a song recording, we can determine the syllable type, but the repetition rate, as I show you here. So the message here is that vocal behavior in canaries presents well-defined rhythmic features on different temporal scales. And since I'll be talking about rhythms in behavior and neural oscillations, I wanted to briefly address previous findings. In humans, for instance, MEG studies have shown that while passively listening to speech, frontal speech motor-related areas synchronize to the speech envelope. On the other hand, in singing zebra finches, oscillations on the local field potentials at HPC, which is analog to motor cortex, shows a 30 Hz rhythm that is compatible with the rate of changes in syllable structure. And so the main question that I will address in this talk is whether a similar phenomenon is present when the bird is listening to its own song. To frame this study, here is a schematic of a birdsong's brain, which presents well-described neural circuitry for the production, maintenance, and auditory perception of song. A songbird brain presents areas of higher neural density, which are called nuclei, that roughly have between 10,000, the tenth of thousands or hundreds of thousands of neurons. From this, nucleus HVC is a sensory motor teleencephalic nucleus that is required for singing and that also receives highly processed auditory inputs. And so, for that reason, it was our target to study the auditory representations of learned vocal behavior, such as the song. To address this, we conducted extracellular recordings on nucleus HVC of anesthetized canaries while presenting the auditory stimuli. We presented 20 repetitions of the song while recording the activity at HVC with a 32-channel linear silicon probes. Here is a segment of the raw data that we recorded. And from this raw data, we can separate the two main components, which are the local field potentials, which are the low frequency components of the data, and the spiking activity, which is the high frequency component of the data, and study them independently. So here I have the data from a typical experimental protocol. On top is the song that was presented to the anesthetized bird. In the middle, we have a schematic of the neural probe used, and the boxes on the left and right represent the activity on each probe channel. On the left, we have multi-unit activity histograms, which were computed by thresholding the spiking activity that I showed you before. And on the right, we have the trial average local field potentials for each channel. And so what we see here is that when looking at a global scale, we see that activity is similar at different depths in the probe, but there are changes in amplitude. So we have similar activity on the depth of HVC. Over here, we have quantified an amplitude-weighted cross-correlation matrix for the activity on all probe channels, which shows that the LFPs at HVC co-localize with an increase in neuronal activity, which was an important characterization of the responses, since only recent studies have addressed local field potentials and their neural correlates in songbirds. So this was an important characterization for us to show this co-localization. If we now look at the responses with a little bit more detail, here I have a song spectrogram of a song that was presented to one bird, and on top, the measured syllabic repetition rate for each song phrase. Here we have the broadband local field potential for the maximum amplitude channel on the probe. And as you can see here, there are certain oscillations on some segments during some segments of the song. And if one quantifies that oscillation frequency on the different song segments, there is a correspondence between the oscillation that is present and the syllabic repetition rate. And this happens not for all song phrases, but when oscillations appear on the local field potential, they are synchronized to the syllabic repetition rate. So if we look now on a phrasal scale with even more detail, what we have here is just a segment, this is a phrase of those P0 phrases that I told you before, which are the longest syllables in canary song. And each red trace here corresponds to the trial average local field potential from the maximum amplitude channel on different sites on HVC. Since these are experiments on anesthetized bird, we can do penetrations on different sites within the nucleus. And as you can see, these slow oscillations following the syllabic structure are present on different sites and even bilaterally. The vertical lines that I show you here are just marking these minima of the local field potentials as a relevant feature on the response. And here we have a histogram where we have measured delta t, which is the time of occurrence of this minima on the LFP with respect to syllable onset. And since these P0 syllables are ubiquitous on canary song repertoire, we can group data from different animals. Here we have data from 3 birds, and events here are these minima on the local field potentials. And we can see that data group together quite nicely, and you have a probability of finding an LFP minima on the syllables way before the first half of the syllable. Normalized time here and on the following slides means that we have cut between the syllable onset and the next, and are just normalizing the times between 0 and 1. We can do the same for P2 syllables. In this case, we only have data responses from 1 bird, but you can see that on different sites, we have oscillations that are linked to the syllabic rate. And also for the P1 syllables, which are a little bit shorter, you can see here that the syllabic period is about 55 milliseconds. And you can see that we have these oscillations following or synchronizing to the syllabic rate. And here we also have data from 3 birds and several of these minima events. And you can see that they group, in this case, towards syllabic offset. So if we group together all responses from all the phrases that we have responses on our experiments, we see that the local field potential average frequency and the syllabic rate are linearly related by the identity linear function. So, and this spans ranges for 3 of the 4 syllable types that I discussed before. And just to wrap up, so I showed you all of the responses we studied from the local field potentials, but I also told you that it was important to characterize the correspondence between the local field potentials and the multi-unit activity, since only recently local field potentials have started to be addressed on Songbird studies. And so, as you can see here, for P0, P2, and P1 syllables, they are ordered by descending duration here. You can see that we have a large correlation between the multi-unit activity histograms in response to P0 syllables and to the local field potentials. So to wrap up, I have showed you that auditory responses to the bird-song song in nucleus HVC present oscillations that are synchronized to the syllabic rate, and that HVC local field potentials capture synchronized neural events in the nucleus. Especially for P0 syllables, which are the longest syllables of different birds, LFP minima and multi-unit activity maxima correlate quite nicely. And just as a comment, this seems to be an entrainment. There seems to be an entrainment to song rhythms, which shares some similarities to what is found on speech processing areas in humans. So thank you.
Thank you, San Diego. That was a great talk. I'm pretty sure that was exciting to have all these songers in your research. Great. I have one question from Jonathan that I believe you have recording, but do you have a HVC recording from song production?
So these experiments that I showed you here, they are just from anesthetized experiments, but at the lab we also record from singing canaries. That's a work that is led, it's the main project of a PhD student at the lab, Cecilia Herbert. She gave a short talk yesterday, but I don't know if you shared the link to the article that I sent you. But
yes, I will share on the chat. Yes,
that's a recent article where Cecilia is first author. I am a co-author of that study, and we show recordings on HVC of singing canaries there.
So I sent the paper that you are talking. Also, all the talks are recording. So if you want to rewatch again some of the talk from yesterday or the days, you can just go back to the live stream in the YouTube channel. And Jonathan also asked again that do you differentiate between neural activities suggesting entertainment and purely stimulus-driven activity?
So it could be that these responses are stimulus-driven. HVC is known to have these selective responses to the bird's own song, but we are studying it. Whether it is entrainment or not, we still don't know. These are just recent findings of these oscillations that were not previously described for canary song.
Yes, thank you, John. Thank you, San Diego. That was great talk, and thank you all the attendees and panelists. That was a great session, and I hope that you had a great time already in NMC3. But there are 3-4 days to go. Hope to see you again around, and have a nice day. Goodbye, everybody.
Goodbye. Thank you.
We are live. I think I'm going to start. So hello, welcome everyone. Welcome to room 9, overall theme, cognition, motivation and emotion. I'm the MC today. My name is Gunnar Schmidtmann. I'm from the University of Plymouth in the UK. And today I'm joined by 3 amazing speakers, and we are really looking forward to a very nice interactive talk session. And I want to emphasize this is an interactive talk session. So this will be a approximately 5-minute talk, and then followed by an approximately 10-minute discussion. So I would really encourage the audience, the attendees, to ask as many questions as possible. You can use the question and answer function. There's also a way that we can actually ask these questions by just talking. Yeah, so there's a way to do that too. Just let us know. And first, I'd like to maybe introduce the first speaker, which is Kate from Queen's University in Kingston, Ontario. She's a PhD student in psychology, and she's interested in behavioral neuroscience. Yeah, Kate, please go ahead and let us know what you have done.
Great, thank you. Okay. So hi, everyone. My name is Kate, and I'm a PhD student, yes, at Queen's University, and I will be presenting part of my master's work exploring the role of oxytocin on rats' anxiety-related behaviors. So oxytocin is a naturally occurring peptide hormone that's produced in the paraventricular and supraoptic nuclei of the hypothalamus, where it then is projects to the posterior pituitary for release into the bloodstream. It's commonly known as the cuddle hormone for its roles in reproduction and social bonding. However, oxytocin-producing cells also send axons to various regions implicated in behavioral defense and anxiety, such as the amygdala and the lateral septum. And so it's not surprising then that oxytocin also has anxiety-reducing properties. And one of the cool things about this system is that it is self-regulating, which means that higher levels of oxytocin release lead to higher levels of oxytocin synthesis. So rats are very social animals, and we see that housing with other rats has been shown to reduce their anxiety-related behaviors. And this suggests that social housing may have anxiolytic-like effects, which has been coined social buffering. Furthermore, it's likely that social buffering is regulated, at least in part, by oxytocin. Specifically, social conduct has been shown to upregulate the oxytocin system, and this could be because tactile stimulation increases the release of oxytocin, and pair-housed rats engage in a lot of tactile stimulation. In fact, 2 studies have even shown oxytocin expression to be increased in socially housed versus single-housed rodents in the paraventricular nucleus of the hypothalamus. And so part of the goal of my experiment was to see if I could replicate these findings. So the objective of my experiments were twofold. The first was to examine the effects of social housing on rats' anxiety-related behaviors. And the second was to see if these effects map onto changes in the oxytocin system of the brain. So 10 rats were randomly assigned to be single-housed and 10 rats were pair-housed for 5 weeks. And then at the beginning of the 6-week behavioral testing commenced with the elevated plus maze and the novelty-induced suppression of feeding test, or NSFT, which I'll explain in a little bit more detail. And then the brains were harvested and processed for oxytocin labeling. So the elevated plus maze contains 2 open arms and 2 arms that are enclosed by walls. Rats tend to prefer to stay in the closed arms where they're presumably more protected. And so we measure anxiety as the amount of open arm exploration. And given my hypothesis that social housing should reduce anxiety, the pair-housed rats should exhibit more open arm exploration than our single-housed rats. Although it didn't reach significance, the results did point in the right direction, with our pair-housed animals, the light gray bars, displaying slightly higher percentage of open arm time than our single-housed animals, the dark gray bar. So the novelty-induced suppression of feeding test taps into rats' natural dislike of novelty. So for 4 days, the rats are removed from their home cage and put into a novel cage before being given a palatable snack, in this case, a graham cracker. It's the same procedure for the single-housed rats. They're also removed from their home cage and put into a novel cage for 4 days. And so anxiety is measured as the latency, or the time to start eating the cracker. If social housing is anxiety reducing, then we would expect that the day with the most novelty, which is day 1, should show the greatest group differences in latencies, and that as the rats become familiar with the snack, their latencies should end up being indistinguishable from each other over time. So we did find a significant main effect of housing, such that our pair-housed animals, the light gray bar, began consuming the snack faster than our single-housed animals. And we also found a significant main effect of day, but no housing by day interaction, which indicates that the latencies decreased across days for both groups. And even though there was no interaction, it was still of interest to look at the behavior for individual days. And you can see here that the largest group difference in latencies was on day 2, not on day 1, as we predicted. I then harvested the brains and performed immunohistochemistry in order to analyze oxytocin labeling in the hypothalamus. And so this is a picture of the paraventricular nucleus of a pair-housed rat with oxytocin stained cells. And so pair-housed rats had a higher number of oxytocin positive cells in the ventral parvocellular region of the PBN. As well, we found that as the number of oxytocin positive cells increased in 3 different regions of the PBN, latency to begin eating that cracker on day 1 of the NSFT decreased, which supports the idea of oxytocin having anxiolytic-like effects. And so what I've shown today is that social housing decreased anxiety-related responding, albeit primarily in the NSFT, and that social buffering upregulated the oxytocin system. And that concludes my presentation. And I do have discussion questions prepared, but I'd be happy to answer any questions anyone might have. Thank you.
Well, thank you so much, Kate. Thanks for the really good presentation. Of course, now we'll be quite interested in the panel or as well as the audience to ask as many questions as possible. So please feel free to use the question and answer, the Q&A box, or just get in touch with us if you have any questions. Are there any questions?
So we can just unmute ourselves?
Yes, please.
Okay. Hi, Kate. I think like one of the most challenging thing here is to establish the relationship between the oxytocin and the anxiety behavior, right? Because the major experimental control is that the one group of animals housed together and another was individually housed. So there could be like so many more things changed besides the oxytocin level, right? So it's more like a correlation between the oxytocin expression and the anxiety level. So what do you think you can further strengthen the correlation or specificity about the oxytocin and anxiety? Like,
right, yeah, so you're absolutely correct that this is correlational, and just because oxytocin seemed to be increased in the pair-housed animals, we can't know for sure if that is what is affecting anxiety. That being said, there is research that have done more direct comparisons between the two, and they've found that oxytocin is increased, for example, it's increased in response to social contact between mother rat and pup dyads. And that's meaning oxytocin's released, or there's higher plasma concentrations of oxytocin in the pups as well as the mothers, and as well as there's a high release of, or not high release, there's a release of oxytocin in the PBN in the pups. And so with that sort of information, we can then be a little bit more confident that the two are connected. Yeah, so with that being said, sorry, did I answer your question?
yeah, I think, yeah, but I think still this is a very challenging question, because it's most of the time it's really hard to move from correlation to causal effect.
Mm-hmm.
Yeah, because this definitely requires more like experiments, like for example, like in vivo control of the neural activity and see the outcome.
Is there pharmacological blockers of oxytocin that you could use maybe?
Yes, so actually I ran another experiment in which I tested the effects of direct oxytocin infusion in the brains of rats on their anxiety-related behavior. So that was actually the second part of this experiment, which I haven't presented today. And there we did see that when oxytocin was infused directly into the lateral septum, which is a region implicated in behavioral defense in animals, we saw it decrease their defensiveness in the elevated plasmase. So the same test that I've shown here today. And so with that work, we meant to do also an antagonism study, but logistics weren't right. Although other researchers have been able to do oxytocin antagonism studies, not with the, actually, yes, sorry, two did them with the oxytocin antagonists directly into the lateral septum, although they were done in mice. And they found a decrease in defensive responding, a decrease in anxiety-related behavior. Aside from that, though, we also see oxytocin antagonists being used in the medial prefrontal cortex, the periaqueductal gray, which is a center for pain, and the paraventricular nucleus of the hypothalamus, and all of those, with those antagonist studies, we're able to localize the effects of oxytocin in those regions implicated in anxiety.
Mm-hmm. Oh, I have another question, is that because you are focused on the paraventricular nucleus, the PVN, which is definitely very important for anxiety behavior. And what we know is that activating these neurons would cause anxiety, right? So, and the majority of the PVN neurons are excitatory because glutamatergic. So I want to know if you have any idea whether these oxytocin positive neurons are also glutamatergic neurons, and what's the effect at cellular level with the oxytocin have on these PVN neurons? Would they inhibit this group of neurons, or would they increase activity of these neurons?
Right, that's a really good question. In my research, I didn't find anything about for example, the co-localization of oxytocin on glutamatergic neurons. That being said, though, we know oxytocin is co-localized on neurons that produce CRH, or corticotrophin releasing hormone. We know in the paraventricular nucleus, we also know that oxytocin is co-localized on neurons that produce vasopressin, which works in direct opposition to oxytocin. So if oxytocin is works to reduce anxiety-related behaviors, vasopressin works to increase them. So that being said, you're right, oxytocin has it's predicted to have inhibitory effects on like CRH release and vasopressin release. But yeah, I wouldn't know its exact connection with glutaminergic neurons.
Thank you. There are some questions here in the audience. There's a question from Billy Lau. Billy, do you want to ask the question yourself or do you want me to read it? Billy,
Yeah, no, I was actually introduced to the Zoom. I thought my Zoom crashed, but I guess I'm in. I'll ask the question. It was a nice talk. I was just wondering if this is an anxiety-related behavior. Have you looked at other anxiety-related markers like cortisol or cortical releasing hormone, CRH?
That's a really good question. So in this particular study, we didn't look at other stress-related hormone release. Again, other research has shown that with an increase in oxytocin is correlated with a decrease in corticosterone levels. It's also correlated with a decrease in CHR release from the paraventricular nucleus. So with that being said, I would suspect that if I was able to test other stress hormones, and we would see the same sort of correlation with that, an increase in oxytocin, there would be a decrease in these other stress-related hormones. Unfortunately, though, no, that wasn't something we were able to carry out in this experiment.
Could I ask another question?
Absolutely.
All right. Does it matter if it is same-sex pairing or opposite pairing to see this kind of effect?
That's a super interesting question, because we do see especially because oxytocin is so heavily implicated in social bonding, specifically a lot of the oxytocin research is actually done in prairie voles, and that's because prairie voles are socially monogamous creatures. They form one bond with
I lost the sound.
Oh dear, there seemed to be a technical problem with Kate's internet connection, I suppose. Let's see if it will recover in the next few seconds. Otherwise, we probably have to then go to the second presentation. Let's wait maybe a minute or so.
Sure.
Well, I suppose the internet connection. Oh, what's going on now? The internet connection is now down completely. I'm afraid we have to continue then. However, I would encourage you to just contact Kate personally to just, support her research, ask her more questions. Her email address, I think, is available from the abstract book. Okay, let's go to the second presentation. The second presentation is given by Gnwt Zhang, and he is currently at the University of Southern California. And yeah, Guang, we're quite excited to hear about your research.
Thanks. So hello, everyone. Am I good? Sounds good. Okay. So today I'm so happy to be here to introduce part of my work regarding how the brain transmits aversive signals, sensory signals into negative emotion through a specific pathway. So because we met for the first time, I would like to introduce a bit about my research interest. So when I was a PhD student, I'm focused on the sound information processing in the auditory cortex. And during the last year of my PhD program, I reported a pathway that can transfer high intensity noise into the brain. So that's a very specific pathway. So during my postdoc training, I continued to investigate the function of this pathway. So one of the key structures is in the basal forebrain. It's called the medial septum. And we want to know that what's the neuronal response properties of these medial septum neurons. So we tried aversive sensory stimulation. Why we called it aversive sensory stimulation? Because here is the two-chamber behavior test, and we placed the animal. The animal can run freely on the two-chamber, and then we placed a noise generator and air blower, and we can see that animals spend much less time on the right side. So we can see the animal like dislike this kind of sensory stimulation. They try to avoid the presence of the stimulation. So we define these stimulations as aversive to the animal. And we do extracellular recording from the medial septum, and we found that these neurons can respond quite well to these two physical stimulations. So we see that from information perspective, the aversive sensory information can be stored or encoded by the medial septum neurons. But we want to know more about whether this information would be used by the animal to generate a specific type of behavior. Then what we did is that we developed a real-time feedback control of neural activity. So we know that physical stimulation would induce activities in the medial septum. And then we designed this two-chamber test. Whenever the animal entered the right side, the animal going to receive the light stimulation. The left side, the animal going to receive the LED stimulation. The LED stimulation itself is harmless, so we coupled it with the optogenetic method. So the light stimulation would induce the influx of the iron. So it would in turn activate the neurons that express these channels. So we first of all, we tested wild type animal, the lung cell type specific activating of these neurons in the medial septum would cause the animal to avoid the light stimulation. And we know that medial septum has mixed cell types. The three major cell types are excitatory neurons, the big glutamatergic, and the inhibitory neurons they get too, and the cholinergic neurons. So we like one by one tested each cell type, and we found that only the glutamatergic neurons would induce the aversive type of behavior. Then we want to know the downstream, because the medial septum is K-stab, because each specific brain structures will project to a lot of areas. So we here did the same thing. We trace where these glutamatergic neurons go. We find they project to a lot of places like preoptic area, then the hypothalamus and the lateral habenula and hippocampus. And then we can place the optic fibers in each of these downstream targets. So we can manipulate the specific downstream of the medial septum glutamatergic projections. And we found that only when we're activating the exon terminals in the lateral habenula, which is very important in the emotional control and depression disease, things like that, we found the animal shows this type of aversive behavior. Then we confirmed that yes, the lateral habenula would respond to this aversive stimulation as well, but this has been reported before, so we just confirm it. And we want to know whether the medial septum relay this information to the lateral habenula. So we infused the drug to silence the medial septum neuronal activity. We found that after we perfused the drug in medial septum, the evoked responses in the lateral habenula was gone. So which means the medial septum indeed transfer this aversive information to the lateral habenula. Then besides the medial septum can induce aversive type of behavior, it also induced locomotion, because whenever we expose to aversive sensory stimulation, the animal also want to escape from this type of environment. So when we fixed the animal and placed on a running plate treadmill, so we found that whenever we activate these glutamatergic populations, the animal gonna start running. And the running is proportional to the stimulation frequency and the intensity. And we found that the activating the terminals in the preoptic areas are responsible for this type of behavior. Yeah, here's the video. You might, I'm not sure if its quality is good for you. Check. So we just proposed this pathway, the multisensory aversive stimulation. This is like bottom-up sensory stimulation that went to medial septum, and one part went to lateral habenula with induced aversion, and when one part went to the preoptic area that caused locomotion. Yeah, and think because of time limit, I can not elaborate every details of the work. So I think that would be all. Yeah. Any question?
Okay, thank you very much. Yeah, again, I would like to encourage the audience to ask as many questions as you like, and as well as the people in the panel, Kate, and only if you have questions, please feel free to ask.
So how do you think this compares to like other circuits for aversive stimuli? I mean, the one I know of is like, you know, decreasing firing in the VTA, but I'm sure there's other midbrain aversive circuits as well.
Mm-hmm. Yeah, you mean this one compared with the one decreasing the activity in VTA?
Yeah, and are there not other previously known ones?
Oh, there are, but I think the one that suppressed the neural activities in VTA are kind of like a major one, because I think a lot of papers reporting the upstream, the input to the lateral habenula, has reported very similar observations. Besides the medial septum, there's a lateral hypothalamic area, and lateral preoptic area, and the ventral pallidum, they all send projections to the lateral habenula. And then the lateral habenula would in turn, though it's in majority cell types are excitatory, and then it would project it to a cluster of cells just near the ventral tegmental VTA, and then these inhibitory neurons would inhibit the neuron activities in VTA. So I think this is just like one type of upstream that would, in the end, inhibit the activities in VTA.
And then from the other direction, like medial septum is also known for like hippocampal theta. So how does that relate to aversive?
Actually, we have tested the stimulation because medial septum projected hippocampus. So we placed the optic fiber to activate these projections. However, we did not see like a acute aversive behavior for this animal. So that one may be more important for learning and memory, consolidation, things like that. But this is more like acute avoidance behavior, because whenever you activate this population, the animal going to start running and going to show this aversive behavior. There's a paper reporting that the medial septum generate the theta oscillation to hippocampus with it, maybe encoded the locomotion information and relay the information to the hippocampus. Yeah.
Very interesting.
Mm-hmm.
Okay, thanks very much. We've got a couple of questions here from the audience. Sandeep Sharma, would you like to ask your question directly to the speaker, or do you want me to read it? Okay, so I'm going to read it then. So Sandeep Sharma asks, can you share information on how you estimated, how did you estimate the speed in 2-chamber in real-time video?
Oh, so if you're interested in this part, you can go to, I have a bioarchive paper describing the methods I used. So it's like a machine learning based method to detect the animal's position, and extracted the animal's position from the background. And you can calculate by each frame the moving distance. It's a pixel level moving distance between each frame, and you have to then you can calculate spontaneous speed for each frame. But also, if you want to make it accurate, you have to do a smooth across several frames, because there's going to be a sudden change. So sometimes the speed gonna become incredibly high like that. Yeah, but I think it's a pretty straightforward method. But you have to do the calculation for the pixel and to the real distance.
Sandeep, does that answer your question, or do you want to have to follow up on it? Yeah, okay. Then we've got a second question, and I think you would like to ask the speaker personally.
Yep. Can you hear me?
Yes.
Yeah, sure.
Hi, lovely talk. So I have a question which you partially answered, that projections, the glutamatergic projections from medial septum to hippocampus. So they're involved in motion, you mentioned, right?
Yes.
So is it like ventral hippocampus or the dorsal hippocampus? That's the first question. Because from your slides, it looked like more ventral.
Yeah.
Is that correct?
So here I only show the ventral part, but we also see the projections to the dorsal part. So I think it covers most part of the hippocampus. I think it has been reported before.
Yeah, and the second question was that dorsal, I can understand that that might be sort of involved in motion or movement or space. What do you think these, I mean, this is speculative, of course, what these ventral connections might be involved in from medial septum to hippocampus?
You mean in terms of like navigation, like a space coding thing?
So usually navigation, for navigation, the dorsal hippocampus is involved. And these are more ventral. Yes. So I was wondering if you have sort of some suggestion what these connections might be involved in.
So from our perspective, we have no idea like how these specific like cell type specific projections to the ventral part would have any function, because we haven't done any like direct test on this part yet. But if you talk about the navigation part, I think there's a couple of reports that when you silence the medial septum, the grid cell in the entorhinal cortex, the grid coding responses would be gone. So you cannot, you no longer see these grid cells. And these grid cells are supposedly provide some information for the hippocampus, because these two structures forms a like triangular structure. They both project to each other, the medial septum, hippocampus, and the entorhinal cortex. Yeah.
Can I just sort of continue with further, or should I?
Yeah, I can continue.
Yeah, so that study that you talked about the entorhinal cortex, those are also excitatory projections like glutamatergic projections or cholinergic projections? Because
I think there are both. If you're interested in, we have shown the projections from medial septum, the glutamatergic projections to the medial entorhinal cortex, and that's very strong. You can check the paper before this one. Cool. But there is definitely also cholinergic projections from medial septum to all the cortical areas, not only like entorhinal cortex.
Cool. Thank you very much.
Sure.
Well, thanks very much for the questions. There are no further questions, and I think we now have to go to our third speaker. Our third speaker is Honi Sanders from MIT, and he's going to talk about animal-to-animal variability in hippocampal remapping.
Yes, thank you. Nice to talk to you all. So yeah, I'm going to be talking about animal-to-animal variability in hippocampal remapping. So briefly, hippocampal remapping, there are place cells in the hippocampus. Each place cell corresponds to a particular location in an environment. Fires particularly when the animal's at a particular location, and when you move to another environment, the place fields will either move or stop firing altogether. And this is called hippocampal remapping, because the idea is that there's different maps for different environments. So, the standard way to say this is different room, different map, same room, same map. But recent theory work that I had done discusses how animals actually have to infer environment identity, and they're not actually given directly labels for which environment is which makes it a much more complicated process than you might have thought. And in particular, we had suggested that this might be an explanation for animal-to-animal variability, which had been mentioned in the literature but never previously shown. So what we wanted to do is reanalyze previously collected data in order to demonstrate the existence of animal-to-animal variability. So the data that we were analyzing is from this paper in PNAS from the Moser lab. They recorded the animal it was, the name of the paper is 11 maps for 11 rooms. And basically they trained the animal in a familiar environment, showed the animals 5 novel environments on the first day, and then repeated the first novel environment and repeated the familiar environment. Similarly on the second day, 5 novel rooms, repeated the first novel room from that day, and then repeated the familiar room. And what this protocol allows them and us to do is to compare both in between different environments and for repetitions of the same environment. So here's a single place cell. This is a particularly active place cell, but this is a single place cell in all of these different rooms. You know, nicely you can see that for repetitions of the familiar room, the place field is in the same location, which is what you would expect if the map was the same for repetitions of the same environment. And when you go to a different environment, the place field is in a different location, again, as you would expect if there's different maps for different rooms. And also you have for these immediate repetitions, the same place field in the same environment. And even for these distant repetition, where novel room 1 is repeated at the end of the day, the place field is also the same. So again, this is a very clean cell, very nice, but just to demonstrate, you know, the idea behind remapping. So we can calculate a score called a rate map correlation between different sessions. So for example, if we compare, say, this N2, the first half and the second half, the rate map correlation for that is 0.94. And the rate map correlation for this session versus this session is 0.14. So you'd expect, you know, correlations near 0, the place fields are different. Correlations near 1, the place fields are the same. So we can average these over all cells to see what the average rate map correlation for a particular pair of sessions is. So what we can see, let's focus on this side. So this is the average rate map correlations for repetitions of the familiar room. And you can see there's a wide variability in the amount of remapping that occurs. But what's interesting is that these are colored by animal. And you can see this yellow animal has consistently high correlations. This blue animal and the purple animal have more consistently low correlations. What's interesting is if you look at repetitions of the novel rooms, you also see variability. You also see clustering by animal. And you also see that the animals that have consistently high correlations in for repetitions of the familiar also have high correlations for repetitions of the novel. So we can quantify this in a variety of different possible comparisons for, you know, these sessions. How similar are they in these repeated sessions, and how similar are they in this other set of repeated sessions? And you can see there's a high correlation for all of these different comparisons, meaning that animals that have strong correlations in one set of environments also have strong correlations in another set of environments, and animals that have low correlations in one set of environments also have low correlations in another set of environments. And then what that implies is that animals that have that the maps repeat very strongly for repetitions of environments are consistent with that, whereas animals that have very weak retention of maps for repetitions of environment are also consistent in that. So to summarize, individual animals have different remapping tendencies. These remapping tendencies are consistent within animals for different types of comparisons. And there was a variety of controls that I did, which I can discuss if you're interested. This work was done by Parsa, who was an undergrad working with me, and then my advisors and funding.
Well, thank you very much, Ronnie, for the presentation. We have actually a couple of questions, and the question is, do you want to ask them yourselves or do you want me to read them? We have first up Eleanor Duvel. Okay, then I'm going to read this question. The question is, are all animals of the same age, gender, etc.?
That's an excellent question. They're all of the same gender. I assume they're all of the same age, but I don't have that information. But it's an interesting question because previously it had been shown that age can cause differences in tendency to remap versus not remap. So that's a very good suggestion.
Does that answer your question, Eleanor? Yeah, okay, it does. Very good. The second question is from William Mao. William, do you want to ask the question yourself or do you want me to read it? Okay, I'm going to read it. The question is, how much RMC clustering can be explained by the number of neurons recorded?
So that was actually one of the controls that I did. Let me just pull it up. So this red animal is the animal that had the least number of cells, 25 cells. This blue animal had a much larger number of cells. I subsampled 25 cells from the blue animal, and this is the variety of different RMCs that you would get from those subsamples. So it doesn't look like it's due to the number of cells. I did a similar thing here where I just subsampled 25 times from all these different animals, and it seems that the clustering is maintained even with the subsampling.
William, do you want to follow up? Do you want to ask another question, or does it answer your question? Okay, he says, thank you. And then I've got a question here from Jason Moore. Jason, do you want to ask the question yourself, or do you want me to read it? Okay. Are the correlation coefficients bounded to be positive somehow? Shouldn't the correlation between the two maps with a peak in non-overlapping places ought to be negative, or do all of the zero bins negate that effect?
This is a very, very interesting point. So actually, this is the one that I want to show. So they're not bounded to be positive, but it turns out that the rate map correlations are mostly positive. And I think that the reason for this is because of the fact that the place fields are either in the same location or they're in a different location. But since most of the environment has zero firing, if the place field moves, then most of the environment is still going to have the exact same value for firing rate in most bins. So you can't really get negative correlations very easily since most of the map is just zero in both conditions. Does that make sense? And I'm not 100% sure that that's the reason, but that's my guess.
Jason, does that answer your question? Thanks. We've run into this to that as well. That's what Jason says. We do have more time. It is we have actually officially 5 more minutes to ask questions. If there are no specific questions with respect to Honi's talk, feel free to ask questions about Kate's and Gnwt's presentations? Or if the yeah.
Oh, okay. I have a question for Honey. So I think because this like play cells always attract my attention, because I was wondering like how the animal gonna use the visual information with the like trying to form internal space about the environment. Because do you change the environment? Sometimes it's mostly regarding with the visual guidance, like, right? So how do you think they are related with the change of this visual information? And have you ever tried to like map the play cell in complete darkness? And would they still try to remap?
Okay, I'm going to answer the first question and remind me if I missed the second. So yeah, the information that the hippocampus is getting, the visual information is highly, highly processed. But it's not only visual information that the hippocampus is getting, it's also getting other sources of information. You know, one thing that people talk about a lot is motion information. For example, you know, when the animals move to room, it feels and experiences being moved. And so it knows in a sense that it's in a new room because of that experience of being moved in between. So visual information isn't the only source of information that the animal has. And with respect to like, how does the animal know whether it's different based on the visual information? That's a very, very difficult question. And you get extremely different results depending on the particular protocols. And that's really what I was talking about in the eLife paper, the theory paper that talked about why it is that you get all of these different remapping behaviors for all of these different training protocols. With respect to the darkness, there was a paper, I think by Quirk in the 90s sometime, that actually they turned off the lights and it caused remapping. But it's hard to say whether it was causing remapping because the fact of turning off the light was something significant enough to induce remapping, or because the animal like lost track of where it was. So that, you know, becomes a little bit of a difficult question. Yeah.
Thank you.
I wanted to ask a question to the listeners, if you don't mind, which is, Eleanor had mentioned that age might be playing a factor. Does anybody have any other ideas of what might be playing a role in this?
Eleanor asked a second question in the Q&A, so I will stand in for her for a moment. She also asked, could there be subtle differences in the anatomical locations of the tetrodes, for instance, on the proximal distal axis?
Yes, that's also a, that definitely might be, and there's definitely reason to believe that that might be playing a role. And I also don't have that information. But the one thing that I do find interesting is that the subsampling seems to have a correlation within the animal, suggesting that, which I'm not sure whether that argues for or against that hypothesis, but it suggests that different subsets of cells also have different remapping tendencies, even within an animal. And so on the one hand, you could say that means it's probably not due to anatomical location because these subsamples are being taken uniformly over the different cells that were recorded from that animal. Or you could say that maybe it does play a role because by chance you're getting subsamples that are somewhere along the recording axis. It's an interesting suggestion.
Thank you very much. I'm afraid it is now 4:45, at least here in the UK, and that means we have to, unfortunately, close this room now. However, I would like to thank the speakers for their nice presentations, as well as the audience and the panelists for the lively discussion that we have. And please, for the audience, please get in touch with the with the speakers in order to answer all the questions that you have. There were some more suggestions there. So then I say goodbye and thank you.
How many people are there usually?
I think like 13 to 20 was around the, yeah, like there were people in them. The question asking wasn't always great. So do not take that personally. I think one, I'm gonna try to remind people this at the beginning of the session, but it is, oh, and we're live, so I will.
Don't say anything stupid.
Yeah. I'll just edit all those swear words that I was going to come out with before. But I think people are in this like in-person conference mode where you hold questions till the end of the talk. But because we have such a tight turnaround and you won't interrupt a speaker by typing out your question, just as the thoughts and feelings arise from you, write it in the Q&A. And that way, once the speaker's done, we can get straight into the questions. That's my advice for everybody on internet conference etiquette. But yeah, and we'll get started in 2 minutes.
How many of these have you hosted, Chloe?
Yesterday was obviously the first day, and that was for my trial by fire. I did 3 sessions. Yeah. I'm essentially a Neuromatch veteran now. That ranks you among very high status. So you guys have the privilege of not being my first, so I'm not like, oh, how do we see chat? Okay, so I guess we can get started. Our first speaker is Chris Rogers, who's a postdoc at Columbia University under Randy Bruno. In case he has more shame than me, I'm going to go out and say Chris is looking for a faculty job. So if you have search committees out there, tell them to tune in. And I will post Chris's Twitter and a link to his paper in the chat. Take it away, Chris.
You're muted.
All right, hopefully everybody can hear me now. Thanks for that introduction, Chloe. Let me try to turn on my, oh good, I'm sharing my computer sound. I do have sound for one of these, so hopefully that will work. So I'm interested in how we identify objects in the world around us, how we explore our world, and how we make sense of the objects that we encounter. And so this is something that you and I might use our fingers to do, as in this image on the left, whereas mice and rats might use their whiskers to do a very similar thing. And so to understand how that might work, I developed this shape discrimination task for head-fixed mice. And I used high-speed video to understand how they perform this task. So what you're looking at now is 2 example frames from the high-speed video. The mouse's head is actually out of frame down here on the lower left. And I've tracked the individual whiskers on the face. And these whiskers are the same in basically all mice, and they have names. So I've labeled them here, C1, the long blue one, C2, the green one, and C3, the short red one in the front. And the mouse's job is to make sense of the whisker contacts that he's making on the shape, and use that to determine whether the shape is concave, as you see on the left, or convex, as you see on the right. And so these are the rules that the mouse has to learn. He needs to learn to lick right for convex shapes and to lick left for concave shapes. But I also trained a control group of mice to learn a different task. So this is called the shape detection task as opposed to shape discrimination. During detection, the shapes are the same and the positions and the timing of everything is the same, but the rule is different. These mice have to learn to lick right if either shape is presented, convex or concave, and to lick left on control trials where no shape is presented. So having these two tasks is going to allow me to identify whether the behavioral and neural effects that I see are just due to the shapes or whether they have to do with the task that the mouse is trying to perform on those shapes. And so here's a video of the task. It's been slowed down 7 times, so it actually goes by a lot faster than this. And you can see that I'm tracking the whiskers here. Good tracking was a lot of work to get working, but it was quite important because as you can see, he's making very brief, very fine contacts with the tips of the whiskers on the shape. And we didn't want to miss even a single one of those contacts as time goes by. So you can see that he's touching both of these shapes and relatively complex patterns, and something about this pattern of contacts must be informative about the decision that he's going to make. So I just want to, because this is an interactive talk, I wanted to pause here and see if anybody has any wild speculation or guesses about how you think the mice might be performing the task. What do you think might be different about the whisker contacts between the concave and convex shapes that the mice might be using to make their decision? So if you have any speculation at all, it doesn't matter if it's a crazy idea or even a bad idea. I'm just interested to see what everybody thinks might be happening. So you can type it in the chat, or I don't know if it's possible for you to unmute yourself, maybe not. Oh, there's one suggestion already, great. I'll give it another 30 seconds or something. Oh, hi, Alan, good to see you here. Yeah, a lot of votes for timing so far. Yeah, so if anybody else can think of something and they want to just, oh, we have a plant in the audience. My collaborator knows the answer already. He's just joking. But thank you so much for these for these suggestions. And this is actually exactly what we thought the mice would be doing before we did the experiment. So the discussion of timing or the ordering of the whiskers, these are exactly what we thought might matter and what might differ between the shapes. And I think it's really interesting that you guys picked up on that too. What we found actually is something completely different, and we were really surprised by it, and it took a long time to kind of wrap our heads around it to understand what might be going on. I think one clue that you could see in the video is that the whiskers are actually really tightly synchronized. The different whiskers tend to hit at pretty much exactly the same time. So there ended up not being actually that much information in fine timing or in the ordering, although we certainly thought that there would be. So those are great suggestions. So I'll just skip the sort of mathematical stuff we went through to come up with this idea. But basically we think that the way, or we know now, the way the mice are doing this task is actually by comparing the relative numbers of contacts made by each whisker. So it has more to do with the number of contacts than the timing. And so what I'm plotting here is the behavioral meaning of the contacts made by each whisker, C1, C2, and C3. And so for instance, on the left, during the detection task, all of the bars are positive. And what that means is that contacts made by each of these whiskers means the same thing to the mouse. It always means that there's something there. It doesn't matter which whisker it was. It means that there was something there. And that's what he's trying to do during the detection task, something versus nothing. And so that makes sense. But during discrimination, this is where we see the really interesting difference. And so now in this plot, positive means convex shapes and negative means that the animal thinks it's a concave shape. And so we can see now, to our surprise, that C1 and C3 contacts now have opposite behavioral meaning to the animal. C1 contacts means that it's convex, and C3 contacts means that the animal will think that it's concave. And that's the case even though all three whiskers can touch both of the shapes. There are some slight differences in the relative proportion of the number of contacts that are made, and that's what the animal is using to make his decision. So now I want to show you a video of some spiking data. This is data I collected in somatosensory cortex, S1. You'll see the behavior on the left and the spikes that I recorded on the right, and audio from one example neuron. So the neuron that you can hopefully hear is emitting spikes in response to whisker contacts that are made. Like now. And one last example trial. And now I want to show you a different neuron. You're going to hear audio now from a neuron that cares about whisking. So we did a lot of analysis of these neural data, and I'll skip again the approach, but basically we found that neurons respond to contacts and to the motion of the whiskers and many other things. But I just want to focus on the most important aspect of the neural responses, which is this. So one example neuron is shown on the left, the spikes that it emits in response to contacts made by C1, C2, and C3 whiskers. And you can see that this example neuron responds much more strongly to C1 contacts than to C2 or C3. And turns out this is completely typical of the neurons I recorded during shape discrimination. The bar plot on the right shows that the population as a whole has a very strong bias towards C1 contacts. Now, as an interesting control experiment, we looked in the shape detection animals. And here we found that there was no overall population bias. All the neurons on average responded the same to each of the whiskers. And so this is what we actually would have expected from decades of research in the barrel cortex. And what was really surprising was this bias that we observed during the shape discrimination task specifically. And so I'll just sort of conclude by saying, what might that mean? Why would there be that bias in the response of the neurons? So this is the same plot you've just seen showing the population bias. And if you think back to the first part of the talk, we now have some idea what is the difference between the two different tasks, and that can explain this, this result. So this is the plot that I showed you, which is the behavioral meaning of the contacts. And in this analysis, we didn't know anything about spiking, but this analysis told us to detect convex shapes, you should count up C1 contacts and even subtract away C3 contacts. And what we're seeing in the neural responses is compatible with that, an enhancement of C1 contacts and a relative suppression of C3 contacts. So that leads me to the proposal that S1 is formatting the responses that are coming in a way that makes it easier for a downstream region to read out a behavioral variable of interest, such as shape identity. Now in the future, what I want to do is look at how motion plays into all of this. So a lot of this is in head-fixed animals. And what I want to do in the future is actually to free the mouse, allow him to freely explore stimuli, measure everything about its behavior as it does so, and relate that high dimensional behavioral input to the distributed responses of sensory and motor brain regions. But I want to just finish up now by thanking the people who helped me along the way. Randy Bruno, my PI, Stefano and Ramon are collaborators in the Theory Center here at Columbia, and Christina and Esther, who helped me a lot with behavioral training, my funding sources, and all of you for your attention. And I think we still have a few minutes left, so if anybody would like to ask any questions about anything that wasn't clear or really anything that you're curious about at all, I would be happy to hear from you.
Yeah, we have 4 more minutes for questions, so feel free to either enter them in the chat or the Q&A box. So I will abuse my position as MC here and just go ahead and ask my question. So what's your sort of next steps with this, Chris? Where is this going to go?
Yeah, so actually I'm very interested in exploring this motion, exploratory motion signal more. That's something that a lot of people are seeing these days. We're seeing exploratory motion signals in sensory cortex. It's been reported by many labs, and nobody really knows what it's for. And my hypothesis, or the theory that I want to test, is that it's there for sensory motor interaction. So you need to know how you're moving your body in order to make sense of the sensory information that you have that's coming in. And so the experiments that I have in mind have to do with freely exploring mice who are moving around an arena and coordinating their motion of their head with sensory input that's coming in. And in that kind of behavioral task where a mouse really needs to integrate head motion signals with sensory input, I think that could be a good way to understand a bit more mechanistically what role these motion signals might play in a sensory decision-making computation.
And I don't know if you know this or if this is an obvious question, but is there something analogous to this in humans as well? Like, has anyone found evidence of this in the way we explore our world?
I would say that behaviorally, there are a lot of strong parallels. I think we don't know nearly as much about the neural processing in humans for obvious reasons. But for instance, one thing that I think is very telling that kind of motivated some of my future experiments is, again, the role of head motion. I think it's been very understudied, but if we think about auditory processing, you know, when kids, for instance, have ear infections and they have impaired hearing in one ear, or people with cochlear implants, a behavioral compensatory strategy that's often used is to move the head a little bit, actually. So you can compensate for your lack of spatial input in the damaged ear by acquiring spatial information in a different way, which is through head motion. And so at that same sort of conceptual level, I think humans do something quite similar. Of course, if we have a good model of this in mice, then that would allow us to look at that at a more mechanistic level and potentially to look at some disease models as well.
Awesome. Well, thank you so much, Chris.
Thank you.
We are going to switch gears now and hear from Jan Aru, who is at the University of Tartu in Estonia, and only 2 months ago has started his own lab, though he has some problems with his internet. Hopefully we can
Let's see, let's hope for the best, people. Yeah. Hey, let me try, maybe it works, I hope it works. Thank you for everyone who is here. I mean, I'm really happy to be here. I'm really thankful for people who are here. The backstory here is that I did my master thesis on consciousness and I did my PhD thesis on consciousness. But after that, I got really, really frustrated. And I felt that we are very stuck in consciousness research. So there were theories of consciousness, like integrated information and global neural workspace theory by De Haan. But they were not simply satisfying for me. So I stopped working on consciousness for quite some time. Because for me, what was always more interesting was the neurobiology of consciousness. So anyway, so I went to study the brain and I went into the lab of Matthew Lacum, wizard of studying layer 5 pyramidal neurons. So these are the layer 5 pyramidal neurons. And well, the one fact you should know about them is that these are the key outputs of the cortex. So if the cortex does anything cool, then these neurons have to output it to the other areas, other structures. And complexity of these dendrites. So in, for example, in artificial neural networks, these neurons are often taken as point neurons, so you just sum up all the dendritic activity and that's it. But Matthew is one of these people who would say it's important that these layer 5 pyramidal neurons have these two key compartments, the apical compartment, or the top compartment, and the basal compartment. And it is important because it seems that these compartments do really different things. So especially if you look at the input to this, it seems that the apical compartment receives feedback. It receives top-down information from higher order cortical areas, from higher order thalamus, etc. Whereas the basal compartment receives feed-forward information, bottom-up data. Of course, because in biology, always the structure defines function. So we are very interested in understanding what this function is. And when I went into the lab, I didn't really want to study consciousness, but then working in Matthew's lab showed me his results. And here they then optogenetically stimulated the apical compartments of these layer 5 pyramidal neurons and did it in the conscious state and anesthetized state. And what was astonishing is that when they looked at the spiking responses, then in the conscious state, led to spiking of neurons, whereas this was completely not the case in the unconscious state. So basically, as if some magic happened here along these dendrites that had stopped this same activation. Then they went further to show that this is basically controlled by the higher order thalamus. So at that point I said, okay, I have to get back to consciousness research. And we started to think about what this really means for understanding consciousness. And I mean, the name
So, Jan, I think we have, you cut out for a wee bit there. Keep going though.
Yeah, sorry, I don't know, and it's of course terrible. I hope that some people who are very interested will have another chat, another possibility to discuss these results. Anyway, so I'm talking about the dendritic integration theory that we proposed, and
Oh, yes, we've had a very helpful suggestion of maybe turning your video off will help your internet plug along.
Sorry, so if you cannot listen at all, then you can, of course, we can take questions or do something else if it doesn't work at all.
No, we're hearing you now.
What did you say, Chloe?
Once you turned your video off, it seems to be helping, so keep going.
Two compartments are basically torn apart. And then the point is that during unconscious states, the input can activate these basal compartments, and it might even activate then nothing will happen. This activity will not propagate. I see a lot of people saying something in the chat. Okay. Okay, I hope you can still listen to me. Yeah. In the conscious state, the two compartments are integrated. Groups that basically can maintain some contents in consciousness. So the point is that this relatively simple cellular mechanism can explain why people find that consciousness basically is correlated to and so we believe that our ideas basically explain the results from Marcello Massimini and others, for example, who found that during conscious processing, TMS pulse creates a long, steady response, which is not the case in unconscious states. We really think that this integration between the two streams is important, that it is important to understand consciousness, for understanding consciousness, to understand that here really two different information streams are either integrated or in the unconscious states torn apart. And so this is important for consciousness. In other words, then conscious processing is always modulated by internal variables. And this is what we see on the cellular level, that the apical compartment represents these internal variables. Okay, and then we thought
So we've lost you again, Jan. But I mean, one, you deserve a medal for presenting with internet this bad, and then two, for tackling consciousness on a cellular level. One member of the audience, and I'm sure everybody else would agree, if your slides could be made available. Oh, and we've lost Jan altogether, have we? Yeah, no, he's here still.
So it didn't work at all, or?
So we lost your audio.
You hear nothing what I'm saying.
But if you could make your slides available somehow, people would love to hear. And maybe if we just slim us down to the very bare bones of no slides and no video, you could answer some of these questions in the chat. For instance, Chris asked, and I had similar thoughts, is there meaning to consciousness other than the opposite of the message?
I am really sorry for this. I mean, this is, of course, very terrible also for me.
Well, this is a start of a conversation, not the one shot of it. So no worries at all. And you have quite a lot of questions in the chat. I don't know, can you see them? So one is there another meaning to consciousness other than the opposite of anesthetize? It seems like it should mean something deeper than that, but I'm not sure how else to define it.
Okay, yeah. I don't know if you can hear me now.
Yeah, we can hear you. Mm-hmm.
Oh no.
Yes, give us your working definition of consciousness.
So I guess nobody hears anything what I'm saying. Great. Thanks for the questions. I guess we have to move on. I will type in some answers.
Perfect.
Here.
Okay, yeah. If typing is what works, I could hear you then or just now. So,
okay. I'm back. Really sorry for everyone for wasting their time. I mean, as you can imagine, it's even way worse for me, right? But so we believe that I'm starting from some questions. We believe that the dendritic mechanism really is the key also to understand better how IIT or global neural workspace theory are implemented. So that if one wants to take into account how they are implemented, then this dendritic mechanism helps to understand that.
Awesome. We have one more. How could this theory contribute to IIT or Gnwt in terms of whole brain dynamics?
Yeah, I tried to answer that one just now.
Oh, sorry.
But probably you didn't hear anything.
Sorry.
I'm answering the second question about the dreaming.
Right.
Yes, I mean, I wanted to tell that, but obviously the internet connection didn't allow me to do it at all. So please check the paper that Chloe also posted. The second paper is about dreaming.
That's awesome. And so we have 1 minute. Do you have a good working definition for consciousness? I don't know if you had said that already and I had missed it from a cutout. Okay, we have lost Jan for the final time. I'm so sorry, but his Twitter is in the chat, and so are a couple of his papers. So please check those out and reach out to him because obviously this was a super cool topic, and everybody wants to hear about it. And we're just failed by the internet once again. But we are going to rally and switch gears and hear from Julie Wefeli, who is a PhD student at the University of Montreal underneath Arlene Kahn. And I am going to post a paper of hers. Julie, take it away. You are muted.
Okay. Am I sharing the right screen?
Yes.
Are you guys seeing my presentation or my notes?
Nope, we're seeing your presentation.
Okay, excellent. So thank you for the introduction. So I will be presenting how aging affects attentional window and perception in the periphery. Previous studies have shown that aging is associated with perceptual and visual cognitive declines. For example, we know that in older adults, they have lower contrast and motion detection abilities. As for visual cognitive abilities, we know that they have a smaller attentional window. That is how broadly they can distribute their attention around the fixation point. However, it's unclear whether these declines interact or not. For example, whether a small attentional window could be explained by perceptual changes. Here we tested whether differences in attentional window in aging are related to perceptual declines, such as lower contrast and motion detection in the periphery, or whether these are independent functions. So we tested 14 younger adults and 11 older adults. We excluded all participants who had neurological or psychiatric disorders. And we also excluded all participants who were taking drugs that could affect attention. We also tested them on the Humphrey to make sure that there was no deficits in their visual fields. And we tested participants with the setup that is presented here. So participants will sit in front of a screen with an eye tracker placed in front of them to record their eye movements. And then participants will perform 4 tasks, 1 attentional and 3 perceptual tasks in a semi-randomized order. For our attentional task, we used the pop-out visual search task. And for our perceptual task, we use contrast detection, local motion, and global motion detection task. So for our pop-out visual search task, participants will have to fixate in the center of the screen. Once their gaze was detected by this window here around the fixation point, the trial will begin. And their task was to locate to indicate as quickly as possible whether a target was present among distractors. So the target is this little balloon and the distractors are these little circles. And there were 4 conditions in this task for gaze contingent conditions. In the control condition, we didn't restrict their window, and then we restricted to 25, 20, and 15 degree window size. And these conditions were gaze contingent, which means that we were using online eye position to make sure that they will move their gaze and the window will follow their gaze. So they had to move around across the screen to make sure that they uncover the whole scene and to locate the target or not. And we use those windows to estimate the attentional window size with a polynomial fit of 2 parameters, where the attentional window size is the point of inflection. The rationale behind it is that if the size of the attentional window is smaller than the visible window, then there should be no effect on search times. So as you can see here, for 20, 25, and 30 degree window size, there was no change in search times. But once we hit the 15 degree window size, you see that there was a clear uptake in search times. And that is because the size of the attentional window here was bigger than the size of the visible window. And the point of inflection is indeed here. So it's bigger than the 15 degree window that was visible for this participant. For our perceptual task, we tested them on the contrast detection task, where participants will have to fixate in the center of the screen. A Gabor patch will appear, and they will have to indicate whether the Gabor was oriented horizontal or vertical. And every time their answer was correct, we decreased the contrast of the Gabor patch. And that was to help us establish their detection threshold with a staircase method to reach an 82% correct performance. For the motion detection task, we used the same experimental sequence, but for the local motion, participants had to indicate whether the Gabor patch was moving downward or upward. And every time their answer was correct, we will decrease the presentation time of the Gabor. And then during the global motion detection task, participants were presented with 3 Gabors like this, and they had to indicate whether the movement across the boards was coherent or not. And every time their answer was correct, we decreased their presentation time until, again, we could establish a threshold. And we tested participants at 2 levels of eccentricity, 5 degrees and 10 degrees. So we hypothesized that aging results in a decrease of the attentional window, which can be explained by a decline in visual cognitive processes rather than a decline in perceptual abilities. Based on our hypothesis, we estimate no relationship between attentional window size and perceptual abilities, where these abilities will decline independently in aging. So here for the attentional window size, we looked at search times as a function of our window condition for both younger adults and older adults. For older adults, as you can see in red, they had search times that were significantly higher compared to younger adults, and we could estimate their the mean of their attentional window size here at 19, and for younger adults, it was around 23. And then we distributed those results across all participants for our 2 groups. As you can see, younger adults had significantly bigger attentional window size compared to older adults. And this suggests that aging affects the size of the attentional window used to perform our pop-out visual search task. For our contrast detection task now, participants, as you can see, our older adults had higher contrast thresholds compared to younger adults. So younger adults could detect contrasts at lower levels compared to older adults. And there was also an effect of eccentricity. So contrast thresholds, were lower for 5-degree eccentricity compared to 10-degree eccentricity. For local motion now, for their threshold, as you can see, there was no difference between groups and no difference between eccentricities. And for global motion now, as you can see, there was a clear effect of age. So older adults had higher presentation thresholds compared to younger adults, but this was independent of eccentricity. So they did fairly the same. They performed fairly the same way for this task, whether it was at 5 or 10-degree eccentricity. And finally, we did correlational analysis across all our scores for all of our tasks. And we only found one significant correlation. So only global motion threshold correlated with attentional window size, and only in older adults. So as you can see, it's the moderate correlation that we observed here that is true for both 5-degree eccentricity for the global motion and 10-degree eccentricity for the same task. So older adults who had a small attentional window size had also higher presentation threshold for the global motion, while for those who had a bigger attentional window, they had shorter presentation thresholds. But this relationship was absent for younger adults, and we found no other significant correlation for younger adults. So overall, aging affected the size of the attentional window, contrast perception, as well as global processing, and this is consistent with previous studies. As hypothesized, we found no correlation between contrast or local motion detection thresholds and attentional window size. Our results reveal contrast detection and attentional window size decline as independent processes in aging, while local motion is preserved. Global processing deficits were moderately associated with attentional window size in older adults, most likely because both processes rely on global scene viewing and integration abilities, which are altered in aging. So I'd like to thank my collaborators as well as funding, and thank you for your attention.
Awesome, thank you so much, Julie. So we have a question from Chris, this is very cool and very related to mouse studies on active sensing that I would like to do in the future. Can older adults use eye motion to compensate for decreased attentional window? For instance, making more saccades before making their decision.
Yeah, we found that in another study that we're doing right now. And yeah, and we are explaining this because they have a smaller attentional window, they have to make more eye movement to integrate the whole scene. So yes, totally.
And I don't know if you spoke to this, I didn't catch it if you did. Is there a way of teasing apart, is the poor perception out in the periphery in older adults due to poor processing in general, or to this smaller attentional window, like you're not able to allocate attention out in further eccentricities, and so you don't have the same discrimination ability?
Well, here we showed that they were completely different. So these two abilities were not linked. It could also be that we're not testing far enough, because we tested up to 10 degrees. We could totally get another result if we're testing at 20 degrees, for example. So right now we're testing kind of not that far away from fixation. So it could just be that at this level of eccentricity, we see two different functions, but at higher eccentricities, we could see that they're interacting.
Awesome, thanks. And we have another from the audience. Has this kind of task been used to study psychiatric disorders such as autism spectrum disorder or attention deficit disorder?
If it has, I'm not aware. We did a similar task with optic ataxia people, but we never tested it with other psychiatric populations.
Awesome. Do you think the attentional window would vary at all depending on the target types that you're using, or how targets are differentiated from distractors?
Yeah. We only tested the pop-out visual search task. If we're using serial visual search tasks, for example, I will wager that the attentional window size is smaller, even more than that. Because the processing time will be, the processing that you need to do in this case is much harder.
Mm-hmm.
So I will expect it to result in something like that. The reason why we didn't do it is because older adults had a hard time doing the task. And if we were asking them to do a serial task, I think they will have like completely killed us and refused to do it. So, yeah, it was a long protocol. So we didn't want to add more stuff than was necessary.
Yeah, definitely. It's always balanced with special populations.
Yeah, especially our older adults were 68 and more. So it was really 65 and more. Mm-hmm. So yeah, they had a hard time.
Yeah. I don't know if you're familiar with Alejandro Lleras' lab. Him and Anna Madison do work on these like race models for how information in the periphery is gathered for pop-out search, and it might be like an interesting thing to integrate in this. You know, they don't do aging, so seeing.
Can you repeat their names?
Yeah, Alejandro Lleras, Lleras, and I think he's at Illinois University or University of Illinois, and his former graduate student, Anna Madison. So that brings us up to our time. I just want to say thank you so much to all of our panelists. Really interesting talks. Jan, you just get points for being such a trooper. And thanks so much for our audience for being engaged and awesome. I think we're not kicked out of this Zoom room for another like 5 minutes. So if there are other questions, feel free to type them in the chat. otherwise find these people on Twitter or email them, read their papers. They're doing cool stuff.
Julie, can I ask you a question?
Yeah, sure.
I was wondering about covert attention. So if they had to fixate one target but actually make a decision about a different part of the visual field, would you also expect that the older adults would have a shrunken attention window for that paradigm?
Yeah, because when we're talking about the attentional window, is like you're fixating there, but your attention is distributed there. So it's already covert attention at this point.
Could it reflect just sort of a general limit on how much computational processing they can do? Or do you think it's really specifically like a spatial window?
I think it's, I don't think it's just spatial. I think it's, because if it was just spatial, then you will expect no difference between pop-out and serial or other tasks or other targets. And I don't think that's true, because there are certain, when they're driving, for example, I know there has been some studies that they do better in some environment and not others. So clearly it's not a spatial, it's not strictly spatial. It's more the amount of processing they have to do.
Mm-hmm. Yeah, and I wonder, I mean, they probably drive more slowly too. I'm really interested in these like behavioral compensations for perceptual deficits. So I wonder if they could, for driving specifically, if they could, you know, maybe if they're moving more slowly, the car is moving more slowly, they kind of make the spatial processing easier for them.
That would be interesting to test, yeah.
I'm impressed or happy that they're doing compensatory saccades. A friend of mine works at the Scapins Institute and studies hemiplegia, and where they've lost half the vision in their visual field, and people do not make compensatory head movements, though they are legally allowed to drive in many states in the US.
People don't make compensatory head movements? That's what you said?
Yeah, no, they don't. I mean, you could solve the problem easily by just shifting your gaze, but
it's not
really
natural.
Yeah.
It's not
Sorry,
natural.
I missed that. Was that a special population or was that?
Hemiplegia, like, so it's like strokes right at the chiasm.
Yeah, oh wow. Okay, that's very interesting.
Yeah.
Yeah, it reminds, there's a professor here at Columbia, Daniel Wolpert, who has a kind of a similar behavioral paradigm where as people are searching the field, like it gradually reveals more and more information to them. And he's more interested in how they choose where to look next.
Oh, that's interesting, yeah.
I'll just put the article in the chat right now, Julie, because I think you might be interested in it.
I am.
It's a very different question from what you're doing, but it is kind of a similar idea of revealing the, you know, gaze, closed loop gaze in visual control. Oh, I sent it to the wrong person. There we go. I sent it to the back end host. That's not useful.
All right, we are going to be booted out of here now. But thanks so much, guys. This was awesome.
Thank you, Chris.
Yep. And, thank you, Chloe. You did a really good job. Thank you.
Yeah, thank you.
I'd like to introduce my work on developing a 3D parenting-based multi-electrode array. As we know, the hippocampus is important for episodic memory formation and spatial navigation. However, the mechanism of the hippocampus remains unclear. So large-scale monitoring of the neuronal activities from multiple hippocampus subregions is crucial to the understanding of the roles for each subregions and the underlying mechanisms of memory formation. However, the current state-of-the-art probes such as microwire or silicon electrodes are much stiffer than brain tissue, which largely increase the chance of infection. But for parenting probe, the mechanical mismatch between the tissue and the implants will be largely reduced because of its biocompatibility and flexibility. So it can consistently record high-quality signals over a long period of time. So in our current work, we propose a 3D parenting-based conformal multi-electrode array. it will realize the large-scale, high-density recordings of the hippocampus. So this is a brief introduction of the fabrication process. First, a layer of pyrimidine C will be coated on the wafer. Then we deposit the electrodes and traces on the pyrimidine C. Then a second layer of pyrimidine C is deposited on the top. Then we do the etching, so the MEA will be prepared for use. So in our previous work, a 2D 64-channel pyrimidine probe with 8 shanks was developed and applied to the chronic recording of the CA1 region and CA3 regions. The longest recording lasts almost 1 year. In our current work, a novel 3D pyrimidine probe consisting of 16 shanks was developed. Each shank contains 8 electrodes, so it has 128 electrodes in total. This 3D arrangement increases the density of the neural recording, and the layout of the electrodes closely match the anatomical structures of the hippocampus. So more hippocampal subregions will be covered for comprehensive understanding of the hippocampus function. This is the insertion procedure. First, the MEA will be coated in a thick layer PEG, which acts as a brace to effectively shorten the shanks because the shanks are very flexible. It's need a insertion aid. So during insertion, only the MEA shanks will be contact with the brain. The PEG will be dissolved by the saline during the operation. In the preliminary results, 59 units of neuronal activities were recorded immediately after the implantation. 8 units of CA1 neurons were recorded from 6 shanks, and 51 units of CA3 neurons were recorded from 9 shanks. This is the single unit activities recorded from CA1, and this is from CA3 region. However, look at the histology slides. The shanks were straight before the depth of 2.5 millimeter in the rat brain. However, it began to split after this depth, which was due to the different dissolving rate on the surface of the PEG block. So next step, a novel design of temporary PEG brace or other insertion aid will be investigated to ensure the straightness of the shanks. So in conclusion, this work demonstrates the feasibility of implanting 3D flexible polymer probe to the deep brain structures, and it realized the large-scale recordings of multiple hippocampal subregions by conformal high-density 3D layout. So in the future, it will be used both acutely and chronically for recording from multiple brain regions in behaving rats, at the same time assisting the development of the implantable hippocampal prosthesis. Before the Q&A section, I want to introduce our polymer implantable electrode foundry. It is a workshop funded by the NIH. It will provide tools and trainings for neuroscientists. Our lab and our collaborators will help you design your own polymer electrodes and fabricate the device and train you how to implant it in the animals. If you are interested, please check this website. In addition, this is the website for our lab, and here's my contact information. That's all my presentation. Thank you. Any questions from the audience?
Thank you very much, Wen-Shuang, for the great talk and sticking to the time exactly. So I think now we have good amount of time for the questions. Before I get the questions from the question and answer panel, so I will encourage all the audience to post their questions there. I was wondering, well, basically I have 2 questions. The first is, have you ever, because you talked about like hippocampus and that these are really designed actually to also capture the structure of different substructures of the hippocampus. But are you planning to develop these electrodes in a way that they would be more generalized to the other cortical structures or subcortical structures?
Of course, because we can design any structures we want for the electrodes. Yeah, if you are interested, please check this website. We can provide any kinds of electrodes you want using the Pyranine C or other polymer materials.
Okay, and my second question was about actually, like, if you have done any comparisons, let's say in terms of both quality of the recordings as well as let's say stability of this quality, like over time, like in your chronic implants. Like, have you done any comparisons in some rats or mice that's where you have let's say like other type of like silicon probes or multi-electrode arrays? Have you ever compared like the stability and quality of the recording, let's say, over time?
Yes, in our group, we have done the comparison between different electrodes for the 2D Pyranine probe. But for the 3D, because it's still in development, we haven't done the chronic studies. But in the future, of course, we will do the chronic experiment for the 3D Pyranine probe, which also compared with other like the silicon probe or the microwire. But for the 2D Pyranine probe, the results is really good. The recording signal lasts almost 1 year after implantation, and we still got very high signal to noise ratio. Yeah.
Great, thanks. I had one more question about, because I can imagine for example, I have worked with multi-electrode arrays, and already when they are stiff, but when you have a bunch of electrodes, an array of microelectrodes that you need to insert, you already get a lot of dimpling of the brain, and then you have problems with insertion and so on. So I would imagine, like in this case, that you have these very flexible electrodes, it would be much harder. So, like for example, the tricks we would use was to somehow fixate like the brain surface to the bone, to the sides of the bone, so that would allow us a nicer insertion of the probe. I was imagining if your guys are also using some techniques that would, I saw you have this, what you call the PEG, to, well, so somehow aid, to hold these probes, to these electrodes. But do you apply anything on the brain surface that would make the insertion a bit easier for you, or no?
Actually, no, we didn't put anything on the brain surface. But after the implantation, after the probe is in the target, we will suggest you put some silicone gel to seal the craniotomy window, so it will act as a supporting and protecting for the brain tissue.
True, that's correct. But I mean, when you're doing the insertion, you don't have like dimpling of the brain, like the electrode doesn't push the brain inside and would not insert, like would not penetrate. You don't have this problem.
Actually, for the 2D Pyranine probe, we didn't have this problem. But for the 3D, actually, because the layout is very intensive, it will slightly press the brain. But we feel it's still okay for the brain surface. Yeah. And if you insert the probe very slow, it will minimize the pressure of the brain. Or there's another way: if you insert the probe very fast, use like a motor, it will also decrease the damage of the brain. Yeah.
Thank you. And I have a follow-up question about the density. Like, what is the limit in like technology you are using in terms of, for example, we know the new probes that we're gonna, I think, hear also in our next talk from Amna, like the NeuroPixel probes. The advantage of this very dense probe is that you could really insert a lot of recording size thanks to technology they're using, that would kind of increase the yield of the number of neurons that you could record simultaneously from the same brain regions. What are the limitations of the technology you're using in terms of number of electrodes you could spaced closely to each other?
Okay, it's a good question. The most difficult thing is the insertion, because the probe is very flexible. Our current insertion aid is use a PEG brace, but however, according to our preliminary studies, it couldn't ensure the straightness of the shanks in the brain during the insertion. So the most challenging part will be how to keep the shanks straight in the brain. Yeah, probably we will try other ways like the coating on the surface of the electrodes to instead of using the PEG block to supporting the insertion. Yeah. Like I said, because you increase the density of the shanks, the force inserting into the brain will be less because the area will be bigger. So the insertion will be the most challenging part.
All right, great. Thank you very much, Wen-Sean, for the great talk and answering the questions.
Thank you.
Thank you very much. So with that, I would like to move to our next speaker. First, I'm gonna, on behalf of all the audience, I'm gonna have a round of applause for Wen-Sean. Thank you very much. And for the interest of time, I'm gonna move to our next speaker, which is Amna Lawrence. Amna is a master's student in the lab of Kathleen Coolen at Johns Hopkins University, which gave a very interesting keynote lecture actually earlier today in the conference. Amna will talk about using the state-of-the-art neuropixel probes to record from deep subcortical structures. Stage is yours, Amna.
Thank you so much for the introduction, Wahid. I'm from the Cullen Lab at Johns Hopkins, and I'll be talking about the utility of the neuropixel probes for studying the vestibular nuclei. So the vestibular system basically encodes head motion. The peripheral vestibular system, that is a part of the inner ear, it sends this sensory information about head motion via the regular and the irregular afferents to the vestibular nuclei in the brainstem. The neurons in the vestibular nuclei either project to the cerebral cortex or the cerebellum for self-motion perception, to the abducens nuclei for gaze stabilization, or to the spinal cord for postural control. Just a brief primer on neuropixel probes. As Wahid mentioned, these neuropixel probes are high-density silicon probes that allow us to capture neuronal activity at high spatial and temporal resolution, and allows us to capture the coordinated activity of many neurons. So here on the right, you can see a typical neuropixel probe. And if we blow up its 1 centimeter shank here, it has around 960 sites, out of which 384 can be used at a time for recording. This brings me to the motivation of our study. So gap junctions are critical because they bring about electrical coupling between the neurons. Connexin 36 is a gap junction that is very prevalent in the vestibular nuclei, and we hypothesize that these gap junctions play an important role in population encoding in the vestibular nuclei. And so we aim to investigate this role of theirs by using neuropixel probes. As a first part of our study, we use 3 paradigms: the vestibulo-ocular reflex paradigm, the optokinetic reflex paradigm, and the static eye position paradigm. And during these paradigms, we recorded from the vestibular nuclei of the mouse brain. The neuropixel probe was placed in the brain such that approximately 111 of the neuropixel probe sites were in the medial vestibular nuclei. And here you can see the spike trains of the channels of the neuropixel probe in that area. And some of the regions do show some modulation with the sinusoidal stimulus. We were able to isolate 10 cells, and we used 2 spike sorting algorithms, namely grclust and kilosort2. And it was interesting to see that both of these spike sorting softwares gave us pretty close results. And for the 10 cells that we isolated, they only showed sensitivity to head velocity and no sensitivity to eye position. And here is an example of one of the cells. To compute the baseline firing rate or the bias and the head velocity sensitivity G, we used a least squares linear regression model. Based on our analysis, we saw that the head velocity sensitivity for our cells, the average head velocity sensitivity was around 0.25 spikes per seconds per degrees per second. And the average baseline firing rate was around 18 spikes per second. And these values, they were well within the range of what has been previously reported in the literature for the vestibular nuclei neurons. So as a next step, we would like to find the synchrony between the single units that we recorded. So for a sinusoidal stimulus, we see that sites that are very close to each other versus the sites that are far away from each other, they do show some coordinated activity. And here by synchrony, I mean that they show high correlation in their firing rate at time lags that are as small as or less than 1 millisecond. And we predict that if we z-score transform their firing rate, then the activity of the pairs of neurons, they will be showing a linear relationship. That is, they will be synchronous. And a similar kind of study has been done previously in nucleus prepositus, where people have seen that if two neurons are as far as 300 micrometers apart, they still show synchronous activity, thereby signifying functional connectivity that exists there. And we would like to see the same for vestibular nuclei. So in the end, I would like to conclude by saying that this preliminary study showed us that we were able to successfully record multiple single units from the vestibular nuclei, and we got coordinated simultaneous activity of many single units. And this has paved the way for us to study the synchronous activity of the single units to infer functional connectivity via the gap junctions between the neurons and the vestibular nuclei. And this has important implications in understanding how our brain brings about self-motion perception and at the same time, spinal reflexes. And so in the end, I would just like to thank my mentor, Dr. Kathleen Cullen, as well as all the members of the Cullen Lab for their valuable feedback and support. Thank you.
Thank you very much, Shamna. Great talk. And again, thanks for staying with time. So again, we have a good amount of time for a nice discussion. So again, I encourage all the audience also to post their questions in the Q&A panel and upvote their favorite questions so we can read them. So before that, I want to first ask, I guess one of the important factors, like when you're using this type of electrodes, is like also like depending on which area you're recording from, one of the factors is like density of neurons and then also firing rate properties of neuron that you're recording from. So how much do you think actually like such a dense probe would actually help you to record, like would increase the yield of neurons that you could have from these structures? Like how dense are these brain stem nuclei, these vestibular nuclei that you're looking at?
So this particular region is like approximately 4.5 mm deep and in the mouse brain. And so yeah, we did find that they were pretty helpful in recording multiple units from such deep brain stem structures. But we did realize that if we like kept these neuropixel probes and used them the next day, then there was significant like movement in the tissue, due to which the sites that you were getting the previous day, they were not the same that you got the next day.
All right, thank you. So we have a question from Plonge Rajbaring, if I pronounce correctly. And the question is, can you explain more about the baseline firing rates? Again, I think it's about these vestibular nuclei and their baseline firing properties.
Yeah, sure. So the baseline firing rate is basically the activity that they show in the absence of any kind of modulation. So So yeah, we got a baseline firing rate of approximately, the average was approximately 18 spikes per second. And this was pretty consistent with what has been reported previously. Previous studies have reported a baseline firing rate that was slightly higher than what we got from our study, but that could be attributed to the sorting algorithms that we used.
So you're only looking at the single units, right? So you're not including any multi-units in this
No.
histogram we're looking at. All
Yes.
right, thanks. So our next question is from Dong Song. And the question is, what's the possible physiological function of the synchronies you observe? And does it mean these neurons are carrying redundant information?
And no, that is not the case. So basically, when there is synchronous activity of many neurons, that means that neuronal response becomes more stronger. And looking at the function of the vestibular nuclei, so for attaining perceptual thresholds, for like achieving self-motion perception, we do want more number of neurons to be able to inform the higher regions of the brain. And at the same time, vestibular nuclei is also involved in bringing about spinal reflexes. And so for bringing about spinal reflexes, you have to overcome the inertia of the limbs. And for that, you require synchronous and coordinated activity of many neurons. And so that is what we are trying to investigate here.
All right, thank you. I had one more question, actually. I think one of the advantages of such probes, I mean, being like, as you mentioned yourself in your talk, that you can have up to 300,
84.
84 channels at the same time being recorded. Have you thought about, and I think these channels can be spread out around the very long probe, like it's a 1 millimeter probe, I guess you have, right? And can you design your recordings in a way where you could target like simultaneously from some like cortical region, like some vestibular regions, as well as some subcortical regions? Are you planning to go in these directions, or like to also record not only from one structure, but like some other related structure that might be functionally connected or relevant?
Yeah, not currently. We are not looking at that correctly. We are focusing more on the vestibular nuclei, but yeah, we can potentially look into that in the future.
Okay, and I think another challenge, I would say, that is like when you want to target some like this very deep structures, is like you need to plan, like if you can like easily miss them by very small amount of like angular mistake at the surface of the brain, then you end up being far away from, I don't know how big are these structures, but you could easily miss these nuclei that are sitting very deep in the brain. How do you design your like? Are you using any like mouse atlases or any automatic programs to decide where to insert your probe and with which angles and so on?
So we do use the Allen Brain Mouse Atlas that is shown here. And at the same time, like we do use these, but then in order to make sure that we are hitting the right spot, we like try to move the mouse, like do some passive stimulation to move its head and see whether the neurons are responding to that or not. And so we know that, yeah, we are in the right place.
Okay, so you can really somehow see, like in your live recordings, let's say a band of electrodes that are responsive to those type of stimulations you're doing. Very interesting. And do you also do some, let's say, do you put some markers, like fluorescent markers to let's say, do histology later on to really make sure that your probes are nice? Okay. Okay. Great. So if we don't have any other questions, I would like to thank you again for a very nice talk, and the audience for the next questions. And again, for the interest of time, maybe we can move to our next speaker. Thank you very much, Shamna. So our next speaker is Tianrei Zhang. And she will talk about the effects of chronic intermittent burst stimulation on adult hippocampal neurogenesis. Well, I just want to quickly remind that tomorrow there is a keynote lecture about human neurogenesis. So if you're interested in this topic, by Silvia Capello, also in this NMC conference. And going back to Tian Ray, she's a PhD student in the lab of Jason Snyder and Fidel Villar-Rodriguez at the University of British Columbia. Voilà, stage is yours.
Hi. Thank you for this opportunity for me to give my talk. I'm doing my PhD and this is part of my project. So my PhD focused on looking at non-invasive neurostimulation and its neurobiological effects. And today I'll present a little bit on a newer form of neurostimulation called intermittent theta burst stimulation and how that affects adult hippocampal neurogenesis. So a little bit on the background. Chronic intermittent theta burst stimulation is a newer form of non-invasive stimulation. It's a varied form of TMS, so transcranial magnetic stimulation, but it stimulates the brain, or it stimulates the it delivers the stimulation at a theta frequency, which is about 5 to 10 hertz. And the development of this technique is so new that we're not exactly sure what's underlying its mechanism. So similar forms of non-invasive stimulation, such as the traditional repetitive transcranial magnetic stimulation and electroconvulsive therapy, are known to induce neuroplasticity, including increasing hippocampal volume and enhancing the unique ability of the hippocampus to generate neurons throughout its lifespan. So in the current study, we want to see that if adult hippocampal neurogenesis is also increased by ITBS, and we will use adult hippocampal neurogenesis as a measure of hippocampal plasticity. So because related forms of non-invasive stimulation have increased hippocampal neurogenesis, we hypothesize that we will also see an increase with chronic ITBS. And lastly, there is a known sex difference in the prevalence of psychiatric disorders among males and females, but there is less known about whether different sexes respond to psychiatric intervention similarly. So we used male and female animals in this case and explored whether there are any differences in their response to ITBS. So looking back, this is my timeline. So we give a chronic ITBS protocol, which tries to mirror the clinical chronic ITBS protocol. And a week before the start of the stimulation, we timestamp the birth date of these neurons with a marker called BRDU that later we can use to examine neuronal survival. And we're also using a unique strain of mice that allows us to induce red fluorescent protein labeling throughout its neurons, so we can look at its morphology later on. So we did 10 sessions of ITBS over 20 days, and then we looked at these markers 3 days after the last session of stimulation. So looking at the neurogenesis markers, we looked at BRDU again for neuronal survival. And then we also looked at double cortin, which is a marker for neuronal maturation. So here's a little summary of the results, but the idea is that ITBS by itself didn't have a significant difference on adult hippocampal neurogenesis. However, there seems to be some sex differences. So not in neuronal survival, but if you look at neuronal maturation, as indicated by double cortin DCX, that there is a significant ANOVA two-way interaction between stimulation modality and sex, in that male, there is a greater pool of maturing neurons only in the males after ITBS, but not in females. And this was interesting because this is we don't see actually a patient difference in response to ITBS in either males or females, but mechanistically in the brain there is something different going on. And lastly, we looked at neuronal morphology of these newborn cells to see if ITBS had an effect on their development. So what we found is that, again, overall, there doesn't seem to be a very strong effect of stimulation. We looked at total dendritic lanes, branching, line density, So the only more interesting finding is the spine density, in that if we do it, if we look at and now analyze it by cell, that there is a trend for decreased spine density after ITBS. So ITBS, we are delivering it at an excitatory frequency. So it might be that as a way to protect themselves from neurotoxicity, they would downregulate their spines. And not relating to stimulation, relating to sex difference, we found that female neurons have a shorter total dendritic length. So newborn neurons in females are morphologically different from newborn neurons in males. This isn't affected by either stimulation modality. However, this is a sex difference that I don't think was explored previously in neurogenesis studies. So overall, our conclusion is that ITBS does not primarily affect adult hippocampal neurogenesis as a mode of action. The male and female adult neurogenesis, there are some differences in that the morphology is different at about 3 weeks of age. And there is a small difference in that the population of immature neurons is greater in males after ITBS, but not females. However, overall, it's comparable between the stimulation modalities. So thank you. And if you have any questions, I'm ready to answer any questions you may have.
All right, thank you very much, Tianrei, for the interesting talk. So I think we have our first question from, I'm sorry, my monitors are small and I cannot read the names. Quite difficult, like I have difficulty to read them. Okay, so we have a couple of questions. The first question is from Farouj Rajbariruk. Okay, I think the first time I pronounced not correctly this name. All right, the question is 20 days a long enough time span to measure hippocampus plasticity? And should we increase experimental time?
Right, so 20 days, we're stimulating them at the time of maturity. So for neurons to become mature, they take about 4 weeks. So this is a significant period during their maturation that I think would have an effect. And we can look at further timelines, I suppose, but I would imagine this is enough stimulations that we should begin to see any differences by neurostimulation. It's 10 sessions overall, which is quite a lot, and clinically about 10 sessions, it should have induced like some response in the patients that in the depressive or patients that do receive this treatment. So we think that this is a good starting point. It's true, it's a good point that a longer sessions, a longer protocol might have a more significant effect.
Yeah, it was a little bit my question that like how do you define, let's say, chronic in this sense, like how chronic is chronic? And yeah, so we have a second question from Dong Song, and the question is, have anyone tested similar effects with conventional TMS? Because you mentioned it's a new technique, the intermittent theta burst stimulation, and what about the conventional TMS?
Right, so conventional TMS, repetitive transcranial magnetic stimulation, is known to increase hippocampal neurogenesis. But again, the problems that like there are some problems that some studies used different animal models where they stressed animals before. So in our case, we did not stress the animals. These were naive animals. And we labeled for neurons that were born before stimulation. So we're looking at the effect of ITBS on development rather than the generation of newborn neurons. But yes, traditional RTMS does increase hippocampal neurogenesis. And what we also did that they didn't is that we looked at the sex difference. And traditional studies, they all used male mice or male rats. And if we look at only males, we actually do see, so although it's not significant, that ITBS have a trend of increasing both BRDU and the double corten in this case.
Yeah, actually, that was my follow-up question that, let's say, or maybe it was also Dong's question originally, that have they also looked at this sex differences, like if they're like in the conventional way. And
it's overwhelmingly male mice.
all right.
Yeah.
So I'm quite naive actually in this field, and I was a little bit confused when you were explaining like this, when you use these two different markers like BR, DU, and DCX, and you were explaining that in one case you do not see so much differences, and in the other case, if I get it correctly, you have some more effect in the male compared to female subjects. Could you a little bit elaborate on that, and like how different what are they capturing, these two different markers, and how could you explain like the differences you observed there?
Sure. So BRDU is a DNA analog. So when new cells are generated, like undergo mitosis and their DNA replicate, they would incorporate BRDU. So this is like the cells that were born at the time. So BRDU labels the cells that were born at that time. And in this case, it's about a week before the stimulation. So we track whether there are newborn cells, like the newborn cells born at that time, their survival like will be enhanced by ITBS or not. Double cortin is a microtubule associated protein, and it's expressed in developing or maturing neurons. So they express it for about 4 weeks at the time, and it's not as time sensitive, like it doesn't correspond to a specific time, like BRDU, like the time we we inject it would timestamp like those neurons. Double cortin is a kind of like a continuous wave that indicates how much, like what's the size of the population pool that's maturing at that time. So it's more, I suppose it's more of a gradual change. And we see that although it doesn't enhance, so in our data, we just see that although it doesn't enhance the sort of like specifically the neurons born a week before, like overall there is a greater pool of maturing neurons. So it might have induced some neurogenesis later on, like during the stimulation period, or it could have had some like effect like around that time. That's what I would say. Yeah.
All right, thank you very much again for the great talk. Again,
thank you.
On behalf of all the attendees. All right, and I would like to thank again all our speakers, Wen-Xuan, Amna, Tianrei, and also Cami, that for the technical support of this session, and all the attendees for the participation and interesting questions that they asked. Thank you very much again, everyone, and hope you enjoy the rest of this conference.